which has specialized tissues thought to aid in light interception and absorption, the palisade tissue in the upper leaf implicated in guiding light deeper into the leaf, and the spongy tissue in the bottom of the leaf thought to scatter light and thereby increase light absorption. But our understanding of leaf tissue optics is based largely on 1D models of particular leaf morphology. So now looking at the left side of the slide, here we've used 3D imaging to identify and model both the conventional eye-shaped palisade and conventional spongy tissue with isotropic properties, but also looking down the slide, alternative morphology such as branched or H-shaped palisade cells and honeycomb pattern spongy mesophyll, which we think is actually quite common. We're now using these image data to inform three-dimensional ray tracing studies in COMSOL multiphysics aimed at understanding how these different cell types influence light absorption and ultimately impact photosynthesis. For example, here in the middle of the slide, we find the more common eye-shaped cells increase light absorption by about 4%. And here at the left side of the slide, we have a comparison of a leaf with a honeycomb pattern spongy mesophyll illuminated either from the top or the bottom of the leaf, where we find that illumination from the top increases light absorptance by about 19%. And these models are just a starting point. We plan on making the models more realistic and exploring many questions within the field of leaf tissue optics with these tools. We have 3D leaf data for over 200 plant species. So if you're interested in this type of inquiry, we are looking for collaborators, and please reach out to me. Thank you so much.
Thank you. That's fantastic. Now maybe Olga Rubi Juárez Rivera.
Yes. Welcome. If we pay attention to the photograph of the beetle, we know that it reflects left circular polarized light. Beetles and other beings in nature present beautiful colors thanks to the selective Bragg reflection, produced by the Bioluminescence structure, which can be seen as a helix. Is it possible to mimic the structural color in the laboratory? Yes, using cellulose nanocrystals. This poster is a summary of the recent contribution of our research group. The green rectangle shows how, starting from filter paper, suspensions of cellulose nanocrystals are obtained in the chiral phase, an analog of the Bioluminescence structure. The procedure for preparing films on a glass substrate by shear coating is also shown. Turning now to the yellow rectangle, we see the photographs of a sample where its blue color, polarization effect, and iridescence are evident. The M41 element of the Mueller matrix in the reflection mode of the films confirms these characteristics. This micrograph shows the Bioluminescence structure, which is responsible for its optical properties. Furthermore, with the experimental Mueller matrix in transmission mode, we were able to find the circular and linear birefringence and dichroism of the samples, which are few reported optical properties in this type of cellulose films. Two approaches were used: the analytical inversion of the Mueller matrix, since the sample does not depolarize, and a regression analysis using the Telehen's constitutive equations, both high as can be seen in the graph. Finally, in the blue rectangle, a preliminary study of the color change in a film after being immersed in water is shown. The color shift is due to swelling of the Bioluminescence structure. So this is how our research group is studying the optical properties of glass-supported cellulose nanocrystal films that mimic the structural color of beetles. Thank you.
Thank you very much. Thanks to all of the Flash speakers for such great and on-time talks. Can I now invite Miguel Augusto Castillo, please?
Yeah, okay, sorry. So is this working? Do you see my presentation?
Yeah, we can see your notes as well. So if you make presentation view.
Okay. Just a second. So where are you? Share screen. There we go.
Great,
Thanks.
perfect.
Okay. You see the laser pointer, you see everything, right?
Does that look good to everybody?
That's great.
Yeah. So, just 1 second. Hi, yeah, my name is Hugh Perryman, and I'm doing a PhD at the University of Bristol under Heather Whitney and Ruth Alton. So my work also focuses on iridoplasts in begonia structures. And so, like Miguel said, a lot of this already, but I'm just going to go over it briefly because it's relevant to my talk as well. So They have these multi-layer stacks with dimensions on the order of visible light. So they have this strong interaction with that light, which looks like strong reflectance. That's these sort of blue blobs that you can see. Oh, I've got a mouse as well. No, I don't have a mouse. And interesting light absorption characteristics as well, which I'll come on to. So they are a modified chloroplast. So they have the same building blocks as chloroplasts. And most interesting to me is, like Miguel, the chlorophyll containing thylakoid membranes. So whereas classic mesophyll chloroplasts have these short bundles of stacked thylakoids arranged in fairly random orientations, iridoplasts have their thylakoids extended over the whole length of the body and arranged into these very well-ordered multi-layer stacks. So here the dark bands in the TEM images are the grana, which is where the thylakoid membranes are contained. So what does this do photonically? Like I said a lot of times, and I probably will say a lot of times, it creates a strong reflectance, and we can measure the spectrum of that reflectance of an individual iridoplast. So that little blue blob, again, is an individual iridoplast illuminated in epi-illumination. So we're shining the light at it and looking at the light that reflects back. And if we measure the whole angular range of the reflectance, then still of an individual iridoplast, then we can find, what we do find is blue shift of the reflectance peak. And that can be seen in the figure on the right here. So if you sort of look from 0 degrees on the left of the figure to 35 degrees at the right, you see this reflectance peak slowly sort of drifts up towards shorter wavelengths. So that's a blue shift. And essentially it's iridescence, and iridescence is like the smoking gun of structural color. So we really are, it's really likely that this blue reflectance comes from solely from the structure, this multi-layer stacking. And there's lots of modeling as well to back that up. It also has an interesting effect on the absorption, like Miguel was also talking about. So by modeling these structures using transfer matrix method, we can calculate the absorption, therefore we can calculate the absorption enhancement, which is defined as this ratio of the absorption with the structure present to the absorption without any structure present, but with the same amount of absorbing material. So this gives us an idea of how much the photonic structure is actually altering the absorption profile. These figures are from this paper at the top right. And these calculations were done for blue reflecting iridoplasts in a cross species called Grandis cross pavonina. It was done for blue reflecting iridoplasts because those are basically the most common ones that we see. So according to this modeling, it looks like for blue reflecting iridoplasts, it looks like the green wavelengths are much better absorbed when compared to a body without the photonic structure present. So this species in particular pavonina in the cross species is native to neotropical understory in Malaysia, where the light will be drastically dimmer than the light reaching the canopy, and also enriched in the greener wavelengths of light, or more accurately sort of reduced in the blue and the red wavelengths of light. And the idea is that the leaves in the canopy have absorbed or scattered a lot of the useful light. So this is essentially what prompted the question for my current ongoing experiment, which is, do iridoplasts change their structure based on the available light? So what I'm doing is I'm growing begonia plants under 3 different groups of monochromatic light, so blue, green, and red, and comparing everything about the iridoplasts to those grown in sort of normal light. I have to put massive air quotes here because due to the growth room that I use, the control group is illuminated by fluorescent tube lighting, which is fairly standard lighting to be growing plants under, but it's definitely not what we can call white light. The spectrum can be seen for that as the upper black line on this figure on the left. And it's just very spiky, basically. What we would consider as white light is roughly an even contribution of all of the wavelengths in the visible light spectrum. For example, like the sun. But what am I trying to do? Sorry. So what I'm doing is I'm taking leaves from plants that have grown exclusively in those light environments and imaging them under a few different microscopy techniques. Firstly, a reflectance microscope, which gives us these images of the blue blobs and allows us to measure the spectrum, both normal incidence and over a wide range of angles. Confocal fluorescence imaging allows me to get information about the size and the number density of the iridoplasts, and TEMs are also important for finding out the structural dimensions and things like that. So I'm going to start with the number density and the size and things. So in general, it was much greater for one species, Grandis cross Pavonina, than for this other species, Morandiae. And this can be seen in the figure on the left. This shouldn't be very surprising, really, because the crossed species in GXP, Grandis and Pavonina, were selected because they display a large number of iridoplasts and very reflective iridoplasts, respectively. This was the cross species that was used in the paper that I showed figures from earlier. And it's kind of like the sort of standard blue reflecting iridoplast begonia that we use at the moment. This other species, Morandiae, Begonia Morandiae, was chosen here by me for being a really sort of fast-growing, low-growing creeper style plant with a very large number of leaves, and it also shows iridoplasts. That one is naturally found in shady forests, like most of these shade-adapted begonias, but these are likely not as deep, dark environments as for Pavonina, the species in the cross species, because it's commonly found sort of in ravines or at forest edges, whereas Pavonina is found in sort of deep, dark, complete cover, neotropical understory. So for the Morandiae, it shows no real change in the iridoplast number density, which, along with the lower number density, maybe indicates that it doesn't really rely on its iridoplasts as much as the cross species GXP does. For the cross species GXP, in terms of number density, it is telling that the blue light group stays very similar to the control group, whereas the green and the red show much lower number density. In terms of the iridoplast size, it looks like the GXP doesn't really change that much, but for Morandiae, the iridoplasts are much smaller in the monochromatic light groups than in the control groups, which is interesting. Now for the bit that really interests me, which is the peak reflectance wavelength for individual iridoplasts. So this is what should give us a really good indication of whether the iridoplasts are changing their structure based on the wavelengths of light available to them. So for GXP, we see a really, really tight grouping for the blue light group, around sort of 480 nanometers, which is definitely in the blue. Whereas the green and red are much less clear because they're much more spread out. We do see a bit of a grouping around 500 nanometers for the green light group in GXP, but overall the median line is actually down at 485, so not too far away from, well, definitely in the blue. For Morandiae, we see a bit of a jump to near 500 nanometers for the green light group compared to the control, but apart from that, all of the other groups are just very spread out, and it looked like Morandiae really didn't respond as strongly as GXP here. So the conclusions so far for me are that the crossbreed GXP responded much more strongly than Morandiae to begin with, because it appeared to alter its amount of resources put into the iridoplasts, and Morandiae didn't really seem to do this, apart from reducing the size of its iridoplasts, which could also have been like a plant stress response. Also for GXP, the iridoplasts in the blue light group showed the strongest, sort of clearest structural change, but interestingly, they appeared to be reflecting blue light in a blue light supplied group. So this could mean that they were maybe prioritizing photoprotection. And it was interesting also that the green light did not cause there to be more blue reflecting iridoplasts, even though from the modeling, we think that those would absorb green light most efficiently. I should add that these were all these monochromatic light groups and the control groups were at really, really low light levels. So they were in shade conditions.
You just got a minute now, Hugh.
Yeah, and just basically my last sentence. So to look at these as modified chloroplasts, to bring us back to the beginning, they actually seem to show much less plasticity than classical chloroplasts, which there's quite a lot of papers that show they show drastically different physiologies, even in the same plant between shade leaves and sun leaves in the chloroplasts. So it seems like for iridoplast, this photonic structure is really baked in. And I say the conclusions so far because I've still got to look at all the dimensions and do the modeling and really sort of confirm all these things that I think I've found initially. Yes. I think we've only got 30 seconds left again, so maybe not time for questions here, but I'm happy to answer questions in the chat as well.
Thanks very much, Hugh. That was a really nice talk. Yeah, definitely post questions for Hugh or contact him if you'd like to know more. Now I'd like to introduce Margot Arnould-Petre. Are you here, Margot? Oh. Great, thanks. I can't hear you, sorry. You're already unmuted, so I think it might be a problem with your hardware.
Can you hear me, Adeline?
Yes, that's fantastic. Brilliant.
Oh, okay,
Take it away.
All right. Screen. Can you see everything well?
Yes, that's great. Go for it.
Okay, thank you. So yes, my name is Margot Minju, and I'm doing a PhD at Natural History Museum as part of the ITN BEEP. And I will take you with me underwater today to look at seaweeds and look at what I'm trying to do to understand how structurally colored seaweeds use a specific interaction with light to better manage their light levels. So seaweeds, just a quick introduction because they're quite different from land plants. They are plant-like, but they are not. They have fundamental differences with land plants as, first of all, they live underwater in fairly shallow habitats, but they don't have a vascular system with like the shoots, the roots, and the stems that land plants have. So that's how they're very different. They're very different, among other things. There is a variety of shape and form of seaweeds in every ocean of the world. And as you may know, oceans are major producers of oxygen. Even though phytoplankton is the main producer in the ocean, seaweeds also play a certain role. And one of the key elements of this photosynthetic activity is the pigments, which are chemical compounds that absorb specific wavelengths, which I guess most people here know. And the thing with seaweeds is that pigmentation is one of the main classification, is what is used to classify the seaweeds into 3 different groups. So you have the green algae, you have the brown algae, and the red algae, which is each have a specific pigments that are not always shared. And for example, for the green algae, the chlorophytides, chlorophyll B is the main pigment. Brown algae has a carotenoid, fucoxanthin. The red algae has a pretty particular pigment, which is a phycobilins. So all the seaweeds are classified according to that characteristic pigmentation. But in each of those groups, you have some seaweeds that show, beside having that chemical color, that pigmentation, they show structural color as well. And I'm going to show you some examples very quickly. But this is Cystosera tamaricifolia. It's a brown seaweed, very characteristic with the golden brown color and the very bright turquoise iridescence. And this is a red seaweed. It's contrast crispus. It has the tips of the contrast crispus show iridescence as well, so structural color. Next one. This is another red seaweed. It's called Chondria scintillans, and the iridescence is a bit less conspicuous. As you can see, it's more like some small spots. And we have another Chondria, which is this one, is Chondria caerulescens. It's also a red seaweed. And as you see, it's very, very bright in its iridescence. And as it was back in August, and it was like all the seabed was like this. So there are different structures that cause iridescence in macroalgae. There's a multi-layered structure, which we find in both the red and the brown algae. And there are the iridescent bodies, which we find in the red algae only, and the microfiber arrays, we only find them in the green algae. So all of these have a specific interaction with light, which we can expect to be different. And we know that light is essential for the survival of the organism, and that it would be interesting to look into what kind of different interaction you would have depending on those types of structures, and also like compared to what photosynthesis actually does in terms of interaction with light. And the structural color has been quite extensively studied in terrestrial organisms, but underwater you can expect the constraints to be very different. And for example, a seaweed would not need to have color for visual communication, for sexual reproduction, like for example, a bird would. So the purpose of structural color and the function of structural color in seaweeds still remains largely unexplored. And that's why we're trying to look closer into it. And for now, we're focusing on the rhodophytes, so on the red algae. And we started by looking at the phylogenetic distribution. So all the structurally colored red algae are in 6 different orders, which are like those orders, which all diverge around 477 to 359 million years ago. So not very soon after the Cambrian explosion, which saw the emergence of countless number of new organisms on Earth. So that's one interesting fact. And what we also did was looking into the natural history museum's herbarium collections to find specimens and look into try to take all the data related to the environmental conditions in which they were collected, in which each of those specimens of structurally colored species were collected. And once they're transposed into a map, it looks like there is quite a concentration of structurally colored red species in areas where sunlight is quite intense for long periods of time, even though like this needs to be more extensively studied to try to better assess the impact of sampling bias in terms of like working with collections. Also, like those collections, unfortunately, on the old specimens, we don't have any information on the depth at which it was collected. It was very rare that people recorded that. And it would be interesting to know because light is unevenly distributed in the water column. So it would be interesting to know if structural color could maybe help mitigate that, as we've just seen with the begonia. And so another thing we're trying to do is look at fresh material that we collected on the English coast. And I'm going to try to go a bit faster. So Chandler and his collaborators did some studies on the Chondrus crispus a few years back. So Chondrus crispus is a red algae. It's from the order Jargatinalis, and it has structural color. The structures are the multi-layers. So here you can see in the micrograph the multi-layers. And it has the particularities that it is iridescent only in some of its life history stages. And what we're looking at now is the Chondria species, which are in another order of the red algae, the Ceramiolis. And this is what we have. So both Chondria caerulescens and Chondria scintillans, so they're both from the same genus. They both have those iridescent bodies. But when you look at it more closely under the optical microscope, you see that even though they're from the same genus and they have the same type of structures, they seem very different when you look at it more closely. And the Chondria scintillans has a more scattered iridescence with also a higher variety of colors.
It's 40 seconds now, thanks.
And lastly, just something interesting, because the Chondria caerulescens has, when it's reproducing, the reproductive structures seem to be much more iridescent than the rest of the thallus, even though the rest of the thallus is also iridescent. So yes, thank you very much for your attention. And I have many people to thank for all this, and thank you also to the organization. If you have any questions, I'd be happy to discuss later.
Thank you so much, Margot, and well done for getting through exactly on time, even with the difficulties. Any questions, please put them in the chat. And in the meantime, can I please ask Sverre Tunstad to share screen?
Yes, yeah. Hi.
Great.
Let's see. Start looking okay?
That's great.
Yeah, all right, cool. Hello, everyone. My name is Sverre Aarseth Tunstad. I'm a PhD student at the University of Bristol under the supervision of Heather Whitney in biology and Ian Bull in chemistry. And for this talk, I will talk to you about Cerinthe major purpurescens, which is a plant in the Boraginaceae family with a set of very beautiful blue bracts. So a bract, that is a flower-associated leaf. So it's not the flower itself, which is blue on this image, but it is the leaves. It's a plant that's native to, well, the genus Cerinthe is found all around the Mediterranean, but this plant in particular is closely situated around the Gibraltar. However, it's very popular in the UK, in gardens and such, and that's probably due to this very strong color. What's interesting about this color is that, sorry, can't. I'm pressing the zoom window when I'm actually, there we go. What's interesting about it is that it's created by a very thick layer of wax crystals. So it's about 2.5 micrometers across, and it can be wiped off quite easily by simply rubbing it with your fingers or with a piece of cotton, indicating that it's the wax crystals which actually contribute strongly to making this color. The reason why we're particularly interested in this is because we think it might be a pollination cue. One, because it's color associated with flowers, so that makes a lot of sense. And two, because it's blue, and blue is very strongly associated with bumblebee pollinated plants due to it's resonating quite well with the bumblebee visual system. And while there are plenty of blue flowers out there already, so that's nothing new in itself, what's interesting here is the fact that it might be a case of structural coloration, in particular created by wax crystals, which is something that we do not know anything about so far. So it would be a completely novel kind of method for creating pollination signal. So thought I'd talk to you a little bit about what plant crystals, plant waxes in general are. So they are found on all aerial non-woody surfaces of plants, and they consist of derivatives of long chain fatty acids. So we're talking alkanes and alcohols and esters and so on and so forth. And they're within and on top of the plant cuticle. So they make up the very extremity of the plant in their interaction with the rest of the world. Their main role is preventing desiccation. So the plant cuticle was essential when the plants colonized the land hundreds of millions of years ago. But since then, they've evolved to play a lot of other roles, such as UV screening, making the surfaces super hydrophobic. And they even contribute to catching insects in pitcher plants. And in some cases, the plant waxes manifest as these beautiful crystals. So you see an example here on a cabbage. You can also see them as hollow tubules on blue spruce in many conifers. And in other occasions, you don't see much crystals at all because it's mainly intracuticular waxes and you just see these kind of granules on top. In the case of Cerinthe major, though, you really do see quite a lot of crystals. As I said, it's a very thick layer, so 2.5 micrometers across. And they are the shapes are very weird. We haven't really seen stuff like that a lot before. So we were quite curious about what actually makes up these waxes. So using GCMS, we show that it's delta lactones. So that's a very rare component in plant waxes. It's actually only ever been found in another species in the Cerinthe genus, in Cerinthe minor, which is quite interesting on its own. And what's also very interesting about these wax crystals, this wax component, is that it recrystallizes spontaneously. So you can wash the wax crystals off of the plants with chloroform, and then if you then take this chloroform solution and place it on a glass surface, over the course of a couple of days, it will then recrystallize. So it will start off, as you see on the left-hand side, as this just granular carpet of waxes. And then over the course of the days, it will then start making these crystals, which look a lot like what you see on the Cerinthe plant itself. On the right-hand side, you can see the very earliest growth stages of these crystals. Those are individual crystals caught at different time stages. A similar effect can be seen on the bracts themselves. So if you remove the waxes and then stagger the time you image them, you can observe basically the waxes regenerating. And the outcome of these waxes recrystallizing is what is, in terms of wax crystals, a very, very blue surface. So it's quite often that you see plant waxes which do alter the kind of visual appearance of the plants. And often that has to do with either thick layers of wax crystals which are being used for either UV screening or superhydrophobicity. But in those cases, the waxes make the surface rather white or grey. As you see on the cabbage leaf on the right-hand side, I've pressed like an SEM stub onto it, and you can see that it just makes it grey. Whereas in the case of Cerinthe major, it makes it look very blue to the eye, at least. And if you measure this using a hyperspectral probe, you can tell that it actually is significantly reduced, the reflection in the UV blue region, which is very interesting from a pollination point of view, because bumblebees love that kind of spectrum. They're very interested in what happens there. And if you take these spectra and plot them into bumblebee color space using color vision model in R, you see that the delta S, which is basically how well it resonates. The bigger the number, the more it resonates with them. Is twice as big for the waxy bracts as the non-waxy bracts, which make it very promising in terms of our hypothesis that this was a structural pollination cue, which takes us back to the main question again. So do they actually affect pollination? So to actually figure this out, we did a pollination experiment where we took a bunch of Cerinthe major plants up to the botanical gardens of Bristol, and we simply removed the waxes on half of the bracts and left it on the other half. So we had 2 treatment groups. And then what we did was we just counted the bees that arrived at the flowers. We noted down whether they visited a waxy plant or a non-waxy plant first. So we only counted the first interaction because it's impossible to keep track of all the bumblebees' visits. So what we ended up with was observing on average about 24 bees landing on the waxy bracts per day and half as many on the non-waxy ones. So, and this was a highly significant interaction. So clearly showing that the bumblebees can see this visual change and that they act based on the color. And what's even more interesting is that this difference was actually maintained throughout the duration of the experiment. So they weren't just shocked by this sudden change in coloration and then didn't care anymore. No, they actually used the color throughout the experiment. So yeah, it seems very clearly that it's a pollination signal. Being a biologist, I'm quite interested in evolution. I'm not an evolutionary biologist, but I was quite interested in how this kind of coloration could arise in the first place, because you need the first blocks to actually start evolution on for this color. And I think the way it's done that is by adapting what's probably been a UV screening on the leaves. So you see the same effect of removing waxes on the leaves in the leaves as on the bracts, but the difference is that in the leaves you have a thinner wax layer, and you also have a very strong peak at the green region, so that's the chlorophyll shining through, whereas in the bracts you have this dark underlying pigment acting as a reflective surface. And that's actually something that's seen in many animals already. So you can see it in A. melanotic Stelis J or in the wings of Morphoacris. So it's kind of a case of convergent evolution, one might say, of structural coloration in 2 different kingdoms, which I find quite interesting. So it's probably a evolutionary pathway of the waxes being used as UV screening in the leaves first, and then spontaneously some pigment has risen in the bracts, and then from there on positive selection on thicker wax crystal layers and more pigment. So to conclude, we have delta lactone wax crystals, which are present on both the leaves and on the bracts. But in the bracts, there is a layer of pigment which really accentuates the color effect. This color effect is clearly visible to bumblebees, and removing the color leads to a strong behavioral change. And as a result of this, we conclude that these wax crystals actually make up a pollination signal. And this is the first time, so this is a novel application for wax crystals. And we do not know at all of any other cases, so it's probably not very common. And I'm not sure how relevant it is for the rest of the world, but I think it's a very fun story. And with that, I would like to thank my co-authors on the paper we're hoping to get out. So that's Rox and Karin Chansmu, who will be talking just after me now, Ian Bull and Heather Whitney. And I would also like to thank the rest of you for listening. Thank you very much.
Thank you for that really awesome talk. We have about a minute to the next speaker. Is there any one question that can be answered in a minute? If not, you guys should feel free to ask questions to Sverre via chat or go into one of the coffee rooms. And so with that, I'd like to.
Okay. Thank you all for attending. I'm going to be talking today about my recently completed PhD work on polarized light and host plant foraging in the small white butterfly. Female insects like my study organism, the cabbage white butterfly, must select among potential host plants, and these choices have a large effect on their fitness. This raises the question, what information do insects use to make these choices? It's generally thought that insects use a combination of olfactory, visual, and tactile cues during this process. However, there may be aspects of the host plant gestalt that remain unexamined. Let's look at a possible real-world situation. Now, if you're a butterfly, how do you recognize and land on the cabbages in the background and avoid this weed in the foreground? Olfactory cues probably won't be that helpful, since host plant volatiles are going to be everywhere. So you'll need to rely on visual cues. There's a bit of a color difference here between the plants, but there really isn't that much contrast. But if you were sensitive to polarized light, you could access entirely different information. Here we see that the light from the weed is much more polarized than the background cabbages. Almost 20 years ago, Professor Elmwood Kelber hypothesized that polarized light could be used as a host cue to help distinguish between plants with shiny leaves like this weed and matte leaves like this cabbage. And this question has remained largely unanswered and was the focus of my PhD work. Here you can see a diagram showing the situation in which sunlight is reflecting from the shiny surfaces of the cabbage on the bottom or potato leaves on the top, with these reflections becoming polarized. The associated compass diagrams show the distribution and oscillation directions of the photons composing each light ray. Notice how in the unpolarized sunlight, the distribution is relatively even around the circle. And we can see in the circle diagrams of the reflected light that the distribution of oscillation directions now bulges out in one particular direction. This is because light oscillating parallel to the leaf surface is preferentially reflected. This predominant direction of oscillation is also known as the axis of polarization, or AOP, and is represented here by the direction of the compass needles. Notice how the bulging is greater for the potato reflection than the cabbage reflection. And we can quantify the strength of this bulging using degree of linear polarization, or DOLP. DOLP is a measure of how much of the light is oscillating at or near the AOP, and it's depicted here by the size of the compass needles. Now, we know that different plants reflect different amounts of polarized light, but are insects sensitive to these differences? Now, I'll lead you through the novel display system I developed for use in paired plant image behavioral bioassays. The system consists of an unmodified LCD monitor, and this LCD monitor produce highly polarized light at one AOP, but we could then alter that AOP by rotating the monitor and then counter-rotating the image. Next, we had a quarter wave plate, which can convert linearly polarized light to circular or elliptically polarized light. And unless you're a mantis shrimp, circularly polarized light is going to appear the same as unpolarized light. The strength of this effect depends on the alignment of the axis of polarization of the monitor and the two axes of the quarter wave plate. We also used a mask for the image, so only the portion of the monitor showing the image is visible. All of this was then presented to the responding insects through a set of windows in the choice arena. And this system, because we used conventional LCD monitors, allowed for independent control of AOP, DOLP, color, and intensity. So the first thing we wanted to do was test a non-host potato image with the DOLP typical of potato against a cabbage image with the DOLP typical of cabbage. This bioassay and all other bioassays were tested at an AOP of 90 degrees or with horizontally polarized light unless indicated otherwise. Not surprising here, we see a preference for the cabbage plant. But the nice thing about our monitor setup is we could then decouple all the polarized light cues from all other visual cues by reversing the DOLP between the images. If DOLP is important, we would then expect the preference to shift towards potato. And astonishingly, we found that the preference was completely reversed, with the butterflies being as attracted to the potato image with the lower DOLP as they were to the cabbage image at that same DOLP. This suggested that, at least in the context of the experiment, DOLP was more important than color, shape, or intensity. The next thing we did was investigate what range of DOLP is attracted to the cabbage white. Similar to the last experiment, only one plant with had a DOLP that varied between 10 and 70%, and this was tested against a control image with a DOP typical of cabbage. What we found is that there was an inversion to most other DOPs outside of that typical of cabbage. While we did find that highly polarized images were attractive, this level of polarization is comparatively rare under natural conditions. In an effort to determine if DOP differences are perceived by the butterflies as intensity or as color differences, we presented a series of choices to the butterfly where the treatment image varied in intensity. In these graphs, the intensity is going to be increasing as we move left to right along the x-axis. In the first set of runs, we presented a pure intensity difference with the control and treatment images having identical color and polarization. In these runs, we saw an increase in the preference for the treatment image as its intensity increased. When we presented a color difference between the control and treatment images, in this case, red versus green, in this case, there was only a significant preference for our treatment image when its intensity was 90% of the control image. When the control and treatment image differed in DOP, but not in color, there was only a preference for their treatment image when its intensity was equal to that of the control image. This similar effect of intensity on color and DOP preferences suggested to us that the cabbage white butterfly perceived differences in DOP in a manner similar to their perception of differences in color, albeit in this case, not independent of intensity. As these results suggested the differences are perceived as color, we wanted to determine which photoreceptors were involved. I'll just briefly touch on the polarization-sensitive photoreceptors of the cabbage white. These butterflies have blue, green, and red photoreceptors that are sensitive to vertically, horizontally, and obliquely polarized light. And because they match up so well to these photoreceptors, we can use the red, green, and blue or RGB color channels to dissect this polarized light discrimination. And by recombining only some of them, we can create an image devoid of blue to impair the function of blue photoreceptors, or an image devoid of red to impair the function of the red photoreceptors, or an image devoid of both to impair both photoreceptors. It was our hope that by minimizing the response of the red and or blue photoreceptors, we might impair the polarization discrimination, thereby implying that these photoreceptors are involved. We started with a baseline test with two identical images with all three color channels differing only in their DOP. Similar to previous experiments, we see a strong preference for the less polarized image at both an AOP of 0 and 90 degrees. When we remove the blue channel suppressing the response of the blue photoreceptors, there's no shift at 90 degrees, but we do see a reversal in preference at 0 degrees. This change in preference suggests the blue photoreceptor is involved in DOP discrimination. When we remove the red channel suppressing the response of the red photoreceptors, the preference is reversed at both AOP treatments, suggesting there is also a role for the red photoreceptors. Lastly, we remove both the red and blue color channels suppressing the response of both these photoreceptor types. We still see a significant preference in these 0-degree bioassays. This again suggests that there's involvement for green photoreceptors. All of these experiments taken together suggest a role for all three polarization-sensitive photoreceptor types in DOP discrimination. So in conclusion, polarized differences can be perceived by the cabbage white butterfly. The DOP affects the host preference, with the preference being for plants with a DOP similar to that of cabbage. And the perception of this DOP is affected both by color and intensity and involves multiple photoreceptor classes. These results support the hypothesis of Professor Elmwood Kelber, showing that polarized light can be used as a host plant cue to help distinguish between matte and shiny plants. Previously, olfactory, visual, and tactile host plant cues were known, but with these findings, DOP of plant reflections joins these other cues as an important new dimension of the host plant gestalt. And these results aren't limited to just this one species. It's likely that polarized light cues could be important for many insects, and they may allow for opportunities for improved pest management. And with that, I need to acknowledge all the help and support from the Grease Lab, as well as my funding sources. And with that, I will now take questions.
All right, thank you very much for that interesting talk. Are there any questions? We have
If I may.
Okay, go ahead.
Thanks. Thank you, Adam, for the presentation. It's very interesting. So do you know what the biological system is that butterflies use to visualize different polarizations, and what could the advantages be? So what advantage is there in seeing different polarizations instead of seeing different light colors like we see?
So they both see different colors and polarizations. It's probably a mixed signal. So these channels are affected both by color and polarization. The big benefit would be that when they land, they can visually recognize an inappropriate host from a appropriate host, the surface characteristics like wax layers will be a lot more species specific than color characteristics, because in general, most leaves are just going to be green.
Okay, thanks.
All right, thank you very much. And perfect timing with that. I'd like to invite the next speaker, and any other questions you have, you can chat with Adam. So the next speaker is Audrey Mueller.
Hi guys, I hope you guys can hear me. I'm just going to try to switch over to my presentation.
Great. So if we can have Darrin Schultz, please.
Hello. Everybody see me? I can't see myself actually.
Yeah, you're great.
Okay. Hey everybody. My name is Darrin Schultz. So today I want to talk to you about something that I did with a couple of my colleagues a couple of years ago. I'm the co-first author with Dr. Séverine Martini, who is now in France. So look her up as well, please. So 2 years ago, I'm in the Haddock lab, and we went on a research cruise off of the coast of Big Sur, California, where we dove to the bottom of the ocean around 4,000 meters. We found this field of sponges, and they all had these comb jellyfish on their stalks. And for centuries, people have thought that no deep sea sponges are bioluminescent, or no sponges whatsoever are bioluminescent. But Séverine Martini checked the sponge for bioluminescence. She took it into a dark room where it was like 4 degrees and poked it and saw that it was emitting light. So this was a big discovery, because previously, we thought that no sponges were luminous. And behind me and on this panel, there are pictures of the sponge in white light and the luminescence and red light and the composite. One of our colleagues at MBARI, the Monterey Bay Aquarium Research Institute, Lonnie Lunston, used morphology to find that these are carnivorous sponges, and I did some sequencing to also find that they were carnivorous sponges, and they're closely related to the genus Cladorhizidae. Let's see. So what I did then is I used metagenomics, and I also used some biochemistry to find that these sponges use the selenorzine to emit light, and that the light that they produce is from their own body and it's not from contamination. So we're following up with genomics and biochemistry to elucidate the world's first luminous sponge. Thanks.
Thanks, Darrin. That's great. So if we move to the next one, Laura Bagge.
Can I say something actually?
Sorry, we've got to move on to the next one. We're really tight with time.
We can't see this. We can't see the speakers, Rox.
Oh, okay.
It's fixed on you.
Can you see me?
Yeah, I can see you. Yeah, when you talk, we can see you.
Hold on. Here we go. Okay. All right, everyone. It is beetle time. So today, I am going to introduce you to some open questions that we're working on here at the Air Force Research Lab. that's related to a rare optical phenomenon in scarab beetles. So in nature, it's rare to see examples of circularly polarized light. We humans can only see this if we look through a filter. And plastic 3D glasses are a cheap version of this kind of filter that you can buy and test for yourself. So many scarab beetles, like this metallic beetle in the top picture, are known to only reflect left-handed circularly polarized light, which is why it appears shiny and golden there on the left, but it's dull and without color on the right. Most beetles appear black or brown when left circularly polarized reflectance is blocked by the right-handed analyzer, as you should see here in my Zoom background. So, or check out the picture on the green beetles. This picture won a photo contest. It's so cool because it demonstrates how a mirror changes the direction of polarization reflected back. So now, if you look at the bottom glasses photo, Chrysina resplendens is unique among all the scarab beetles, and that switches handedness, and it reflects both left and right circularly polarized light depending on the wavelength. So we want to know more about how this variation, how much variation there is in this optical signature within this one species, in an attempt to understand whether or not there's any visual function. So some have hypothesized that resplendence reflects both directions because this increases overall reflectivity, which may aid in camouflage. So we want to do more studies in the beetle's natural environment. So ultimately, we're performing optical, electrophysiological, and behavioral tests together to confirm whether or not these beetles have any ability to discriminate this light. So if this brief summary has made you catch the polarization bug, I'd love to work together to collect some gold beetles, maybe in Costa Rica. And so we at FRL are always seeking opportunities to collaborate. Thank you. Okay, I see my slide is finally shared, but hopefully if you were looking at my Zoom background, you were able to see what's going on. Thank you.
Thanks very much, Laura. I'm sorry about that. I wanted people to see your background because I knew it was a video. So if I can get Hannah Weller to present. Thank you.
Cool. So yeah, it's still beetle time. Thanks, Laura. Yeah, so I'm an evolutionary biologist by training, and anytime you're an evolutionary biologist, you have to quantify a phenotype and then measure it in a bunch of different individuals in your clade of interest. So for jewel beetles, this is a huge problem because anyone who's ever tried to quantify a color pattern knows that you kind of immediately run into the problem of like, what do I even measure? What do I quantify? I think most of us sort of take the top route, which is the totally manual. There's a ton of methods to this, right? Audrey Mueller kind of really beautifully illustrated this. The methods usually fall somewhere between super manual and super automated. Super manual, we're usually pretty familiar with. You kind of come up with a bunch of different characters. So here I have some like categorical characters like pattern type, and some continuous characters like maybe your luminance measurement, or maybe you're doing something more sophisticated. But in the end, you end up with kind of a huge gangly character matrix where you have all your species in rows and all your characters in columns. In our case, for the jewel beetles, we ended up with like 50 something characters. And it's great because it's super granular, it's super transparent, but usually you kind of throw your hands in the air and you do a PCA and then you use PC1 as your variable and then you kind of walk away from the whole situation. The other extreme is to go for a neural net, which is, you put all your images in and it gives you out distance scores. And that's what I'm showing in this neighbor joining tree here. It does a great job of organizing your images by how similar everything looks in this really holistic way. And I think this makes biologists really like tense up because it's a black box. Like you put in your images, you've gotten out distance scores, you don't really know how much biological relevance they have. You don't know if something else about the images is getting in the way instead, and that's what it's picking up on. So we ran both of these on the same set of images. And at first we were like, okay, now make them fight. Which one is better? Which one is the correct alternative? And then we sort of realized they're doing different things. The neural net is holistic and maybe kind of a black box. The character matrix is straightforward, but maybe a little bit too granular. We can actually use XGBoost in this case, but a version of a decision tree algorithm to unpack the black box scores using the character matrix. And you can use this to go in between the two and kind of zoom in or zoom out as much as you want. So you can use the similarity scores to decide how important the characters are.
Thanks very much. I'm really sorry. I'm going to have to cut you off there.
No
Thanks. Can I now ask Fabiola Cardoso Delgado to come forward, please?
Yeah, can you hear me? Okay, cool. So my name is Fabiola. I'm a PhD student with Heather Whitney and Tom Oliver at the University of Bristol too. So my research is in Phylogatis rotundifolia. It's another understory plant from Malaysian rainforest that displays blue leaf iridescence, as can be seen in the top left image. Hugh and Miguel have done a bit of the background of how this iridescence is originated in the leaves. So basically, I'm going to go into the characterization that I've done. So I did reflectance light microscopy in these leaves, which allowed us to image and measure the reflectance spectra in vivo of individual iridoplasts. But what I noticed was that when I was doing the measurement, the iridoplast went from bright blue to green, as can be seen in the top right image. So I decided to do 3 consecutive measurements on the same iridoplast to see how this reflectance evolves, basically. And you can see that in a matter of minutes, the spectrum does decrease and then redshifts dramatically. So I wanted to see if this reflectance, basically how reversible and how dynamic it was. So what I did is I performed continuous irradiation for 10 minutes, and then I would measure the spectra every 2 minutes. And then I let the sample relax for 15 minutes under complete darkness and see if I could get the reflectance to recover effectively. So at the bottom right plot, the blue data points are from the light irradiating measurements. So I was constantly irradiating with white light and then measure the how the peak reflectance changed. And you can see that it does redshift over time. And then the dark red ones are when the leaf is readapting to darkness. And you can see that the reflectance does recover slightly over time. It does take longer to recover than it does to redshift, but it does come back after some, like around 10-15 minutes. And then I also wanted to see if these changes were caused only because of the light intensity or if it had some wavelength dependency. So on the bottom left, you see a figure with the absorption spectra of the different pigments in plants. And the thick lines are the different wavelengths that I picked to irradiate and see how the sample behaved under constant illumination on single wavelength. And I think that's it.
Thank you very much, Fabiola. That's fantastic. Finally, for the last flash presentation, can we please have Sierra Weir?
Yes, can you hear me?
Yep, that's great.
Hi. Awesome. I am with the Lore Lab as well, and I'm focusing on the evolutionary implications of color in the primarily iridescent and economically important jewel beetles. While others in my lab, such as Abel Chow, are investigating structural color, I'm focusing on pigmentary color. So a lot of studies have elucidated pigments in groups like birds and butterflies, but surprisingly, we know little to nothing about pigment in beetles, which are the most diverse group of organisms. So I'm researching an irregular charismatic group of pigmented jewel beetles that also produce taxon-specific chemical defense compounds, suggesting their color is a signal of warning or aposematism. They also have a disjunct Gondwanan distribution, suggesting that they have been successfully adapting to a diversification of resources for over 180 million years. So their red, orange, and yellow pigments could reveal important evolutionary processes of color and communication. But first, I had to identify the pigment. So the most informative data for my study are solubility and absorbance, because each pigment class behaves differently in different solvents. So I eliminated the pigment classes of carotenoids and melanins based on their very different solubility and focused on omochromes and pteridines. So in a basic environment, my solutions had some color and an unstable spectra, and in an acidic environment, the solutions were moderately colored with more stable spectra. And these results are consistent with known omochrome behavior. And the shape of the absorbance spectra, as you can see, it looks pretty similar to the characteristic omochrome peaks as well. While I do need more data to confirm these results, but it will either lead to the identification of the omochrome pigment class, or I will need to perform more tests to identify a novel compound. And first, I chose to focus on the Australasian genera, and next I will be investigating the neotropical genera, which may produce different data. So the pigments could be different according to region, or they could be novel pigments altogether. That's all.
Thanks very much. Thanks to all of the flash presenters. And now we'll move straight on to the next talk, which is by Heer van der Kerkhof. Is that here?
Yes, thanks. Share my screen. Can you see my screen?
That's great, yeah.
Okay, so my name is Gea, and I work on structural color in bacterial colonies. So I'm sure that most of you will know what structural color is. We get colors created by interference of light waves coming from visible light that interacts with nanostructures. And it's very widespread across nature and also occurs in bacterial colonies. And in bacterial colonies, it has been found that the coloration comes from the organization of the bacteria cells within the colony. So they organize into crystalline structures that reflect colorful light. And over the past few years, we've learned a lot about the optical mechanisms that are used by these bacteria to create colors. But we don't know much about how these structures are created. And what we specifically don't know about is the biochemical pathways that are used to create this coloration, to create these structures. So this is something we have looked into, and we studied a strain called Flavobacterium IR1. You can see it in this picture here. You see a colony on a black agar background. And if you look at this colony in top view, you see with SEM that they are rod-shaped cells that are lined up nicely next to each other. And in cross-section, you see that they form hexagonally packed crystal lattices. And it is actually this hexagonally packed lattice that creates a 2D photonic crystal that gives it the color. What we found for this strain is that if we grow it on what we define as the standard nutrient condition, when they grow on an artificial seawater agar substrate, we get an iridescent green colony. But if we add to this nutrient mixture now a polysaccharide, a type of sugar called fucoiden, the colonies grow more matte and purple. And this is quite interesting, especially because we found that it is not a response that is just generally comes from adding any kind of polysaccharide or sugar. Because if we add a different type of polysaccharide instead of fucoiden, if we add, for instance, kappa-carrageenan, we don't get the same kind of response. So it seems like they have a very specific response to this fucoiden component. And this is especially interesting because fucoiden is produced by brown algae, and these bacteria, they come from the sea. So this could potentially indicate some kind of link to the function of this kind of coloration. I can think about symbiotic mechanisms, for example. So we studied this in more detail, starting with the characterization of the optics. So these colonies are iridescent, as you see in this video. You see a colony here on a piece of agar that's mounted on the glass slide, and by rotating it, you can see that the color only appears under certain angles. So then we characterize this, this color by a goniometer, where we shine light on the sample at a fixed incident light angle, and then we detect spectra under a range of detection angles, which creates a plot such as this one, for example, where we have the spectra, the wavelength resolved for detection angle here on the x-axis. You see these yellow high intensity reflection spots combined, they are the ones that give the color. And from these type of measurements, we can derive a lot of information on the structural organization of the bacteria. One thing that we can find out is the origin of the color change, which stems from a change in the lattice constant. The lattice constant is indicated here in this sketch, for example, by the parameter D. So it's the interbacterial distance of the cells. And we can measure this distance from by analyzing the optics of the plots. And I don't want to go into too much detail on how this analysis is done here due to time constraints. But what we got from it is that actually between these two different nutrient conditions, there is a 25 nanometer change in the lattice constant, which is enough to create this massive color change. So it's a very sensitive system to very small changes. And in addition to this lattice constant, we could also find the origin of the matte appearance that happens in the phacoidin grown colonies. And the way that we can find this is by looking at the amount of tilted domains in the sample. The way that works is if you look at, for example, in situation number 1, we have a crystalline domain of bacteria, which is aligned quite nicely to the surface of the agar here. And we can expect that the majority of the crystal domains will be aligned to the surface of the agar, because this is sort of the easiest conformation for them. And for this conformation, the light will be reflected under a very specific angle. And so, because this is the majority of the cases, we will expect that the highest intensity reflection peak will correspond to these aligned crystalline domains. However, in another situation, you can also have domains that are not so nicely aligned. They are tilted. And if a domain is tilted, it will also have a tilt in the reflected light angle. So for example, situation number 2 would be reflecting here a little bit to the side. And if you look at these plots, you see that this is actually the case. There are tilted domains because at the high, we have the high reflectance intensity spots. And in the angular space around them, we see that there is also light reflected at different angles, although albeit a lot lower. So what we did is we took the intensity at these 2 different spots, and we took the ratio between these 2 intensities as a measure for the amount of tilted domains we had in the sample. And from that, we found that actually the phacoidin case does have many more tilted domains in that sense than the standard condition, explaining the matte appearance in that sense. So then to further understand the mechanisms that govern this phacoidin specific change, we screened a transposon library for colonies that no longer showed the matte purple color when grown on phacoidin. And what we found was basically from that is mutant strains that all had a disruption in this particular metabolic pathway. So what's displayed here is a polysaccharide metabolic pathway, which acts to partially break down polysaccharides and also to function for the uptake of polysaccharides into the cell across the different membranes. And what we saw is that if we disrupt any part of this pathway, so for example, the Sus-C protein here, which acts to transport polysaccharides across the outer membrane, if we disrupt a transcription factor that is upstream of these green proteins here that you see, or if we just disrupt the inner membrane polysaccharide transporting protein, in all of these cases we lose this matte purple appearance for the colonies that are grown on phacoidin. So basically, it starts more or less ignoring phacoidin, which means that it is this pathway that is involved in this tuning of the color. And this is quite interesting because it means that it is no independent sensory mechanism that happens outside the cell that tunes this color. It's really the uptake and metabolism of this component that changes the color. And what's especially interesting here is that it is a very specific mechanism in the sense that these bacteria strains, this bacteria strain has a multitude of different polysaccharide metabolic pathways, different ones. But it seems like this one is the only one that actually maps to these color changes. So it's a very specific pathway, which shows again that it's not a result from a general polysaccharide metabolism, but really specifically related to this phacoidin compound. So to conclude, we found that the coloration of these bacteria strains can be influenced by the presence of certain polysaccharides, specifically phacoidin. And these phacoidin effects are very specific because there is one key metabolic pathway that is connected to these color changes in the presence of this particular compound. And because phacoidin originates from a certain type of algae, this raises kind of questions about the function of this color change. Because it seems like these strains are used to tune the color when in presence of these algal products. Thank you very much. I'd like to thank the people from the Bio-inspired Photonics group in Cambridge, where I work, and also the people from Hoekmijn, which is a biotech company in the Netherlands that works on the structurally colored bacteria, who have collaborated with us on this project. Thank you very much.
Thanks very much, Gea. There's 50 seconds if anyone has a very quick question. Otherwise, we can move on to the next speaker. Yeah, perhaps put any more questions for Gea in the chat. And now, can we get Warren Francis?
Yep.
Brilliant. If you'd like to share your screen, thanks.
Okay, is that working?
That's great.
Okay, so I guess I'll be beginning some of this section of bioluminescence in the bacterial and bioluminescence, and I'll be talking about a project which began in 2018 through 2019 and was published last year, so it's feeling a bit old, but I'll combine it with some results and observations that are a bit more recent. So compared to the birds and the butterflies, the corals are in a phylum called the Cnidarians, which is probably still familiar to everyone. This is our jellyfish, coral, and sea anemone phylum. And the size of each one of the triangles here of rough phylogeny is indicating the number of species. And the number of blue dots here is indicating the number of bioluminescent species. So of interest, I'm going to be talking mostly about this group off to the left, which is the octocorals, or also called the soft corals. So this includes things like sea fans and sea pens. So things that kind of a very different shape than our normal reef building corals. Many of these can live sort of in soft substrate or in mud, or they're attached to rock. They're typically just filter feeders, so they sort of let their polyps out into the water and just kind of either drag particles that are drifting through or very small prey. And they don't have eyes, but they have photoreceptors. So we want to think about that as we're moving forward into the bioluminescence. So they can make light, they can probably see the intensity of daylight, the intensity of their own light, but they're not making an image. So some of these deep sea ones that we were observing in 2018 have a pretty big range of colors and shapes that are found at a whole bunch of different depths. And like many things in the deep sea, they're not very well studied. Sometimes they're not described, sometimes they're only described, but there's nothing known about the biology beyond the morphology. And then maybe they got dredged up, put into a jar of alcohol, and then shipped to a museum on the opposite side of the planet. So there was pretty much a lot to learn. You bring them up from the deep, they often don't survive very long, so you can't do a good amount of experimentation, but there's some you can do, and this is why much of the result I'm talking about will just be observational. The key ecological question here is what is the function of the light? And related to this would be thoughts about who is watching the light. Now, compared to many of the things which have been discussed already, those who aren't that familiar may think the bioluminescence is a very different phenomenon, but it actually is going to serve most of the same functions as pigmentation in general. You can use this for mate attraction, you can use this for camouflage, or you can use this for warning coloration, a whole bunch of different things. And bioluminescence is going to do the same. The difference is that instead of relying on daylight of basically every other animal on the planet, you're creating your own light, and the intensity and the patterns that you create are what you can use to control who's seeing it or what you want to be understood. The other thing that it actually has in common with bioluminescence and pigmentation is the molecular mechanisms may be very different for different species, but the phenotype may wind up being the same. If we look across animals, we might find a dozen different red pigments, right, that are made by different genes, that are the different chemical structures, but the function of that phenotype of appearing red may actually wind up being similar. So we want to keep this in mind as we move forward and start thinking of these functions. So one example of a shallower species that I was able to study last year comes from the Mediterranean of Panatula, which is the origin of the term of the Panatulacea, which is the sea pens, and another species called Pteroides. So both of these, they're about 20-25 centimeters long, so you can see that the images here, something about the size of your hand, and both of them are bioluminescent in a very bright green. Now, if we do a long exposure with the camera, you can see that the overall outline of the animal and kind of the pattern that it's making. But this sort of belies the actual function. So much more like cephalopods, where many species can dynamically change their color, what we're actually seeing in these species is that there's very rapid waves of light that are moving across the animal. I'll try and show a video of this. It's going to be oriented like this, so that sort of the apex is going to be off to the right and the base of it's going to be off to the left. And so what I'm doing, this is a specimen that we caught and is in the lab, and I'm about to add potassium chloride. And so I hope everyone's able to see these very rapidly propagating waves starting at the base where I added the potassium. So the point that's being stimulated is going to stay stimulated, but then the waves will propagate from there. And here I add more potassium and it does it again. Now I can switch to the other side, so from the apex, and now we see that the waves are actually going in the opposite direction. So that they're very clearly going from the point of stimulation. If you do it in the middle, it goes in both directions. So thinking about what is the function of this very conspicuous and very bright light. So this could be something similar to aposematism, could be like a warning signal, but it also could be something, this was hypothesized 80 years ago, they call a burglar alarm. And so something this conspicuous, to me, this actually looks like a burglar alarm. So here's what this is. The idea is that if you're a very small prey and there's a predator, there's a number of things that predator has to do in order to ultimately make a meal out of you. So first it has to see you. So you can defend against this with camouflage. It has to catch you. Now, if you're very slow and planktonic, the catching part might be pretty much given. And then it has to eat you at the end. So you might have a way of fighting it off once it's basically gotten a hold of you. So in the case of this jellyfish, if it's found and it's caught, that's pretty much the end. So what can it do? Well, it has one way of fighting it off, which is to produce light to signal to something that's substantially larger and meaner that wants to come and eat your predator, and hopefully not eat you. So thinking about that in the context of the sea pens, again, they can't move, they can't see their own, they can't really make out the image of their own light, so they're probably not talking to each other. There's another species that actually lives in the same place in the Mediterranean, but it's a bit shallower, so there's a bit more known about the ecology, and that's one called Veratillum. So just like the other sea pens, it can kind of inflate and contract its body. So when fully contracted, it might be about 10 centimeters long, fully extended about 30 centimeters in the two images on the left. It also produces a green light. I won't show the video, but you can go find it online. But particularly of interest here is when we start thinking about the predators. And there's one predator in particular. It's a nudibranch, so a sea slug called Armina maculata. And what we can see in the photos on the right, so one of them, this Veratillum, it's this orange stalk sticking into the mud. And this sea slug actually has a very similar color and pigmentation pattern of this orange with the white spots. So if it were eating it during the day, it's actually camouflaged on its own prey. If it were eating it at night, this is when it might be relevant as either a warning signal or as a burglar alarm. Now, it's probably not a warning signal, and the reason we know that is because it basically, Armina is a specialist predator. It can't really eat anything else. So you're not going to scare it away with whatever light you make. You're not going to distract it. You're not going to startle it. If it found you, it knows it wants to eat you already, because that's the only thing that it can eat. And so this is why it would be very relevant in this case of trying to signal to a second order predator using light. I had asked a colleague what potentially would eat these nudibranchs, let's say at dusk, and she said probably something like crustaceans. We will look at the visual sensitivity of a number of other crustaceans. This is a crab and a lobster. They seem to have relatively good sensitivity in the green light, which is the color that a lot of these sea pens make. If someone knows of a database, for instance, of visual sensitivity of a whole bunch of animals, let me know. I'd be interested to look into this. So just send me a message of that later. But I would say in the meantime, this is a good candidate of potentially what would be serving as a second order predator. Now, I haven't talked about the deep sea ones much. So these guys are interesting because quite a few of them actually produce a very distinct blue light, but some of them still make green due to the presence of GFP, just like many of these sea pens. So one of them, for instance, from our study came out at the beginning of last year, looks like this. I would say that's very strongly blue. That's a blue as far as I can see. And the wavelengths that are emitted are pretty much monochromatic. so we can talk about colors in this way. Now, there was a set of data compiled by a guy named Woody Hastings 25 years ago. And this actually really makes it look like in different habitats, there's very strong convergence to very few colors. You're either green in a coastal, you want to be green, or you're blue in the open ocean. But the kind of wild card here is what they call the deep sea. So he probably meant benthic deep sea. But it's a much closer split between green and blue. And so when we were wondering, kind of as an afterthought of that study, what potentially could favor the green or blue in the deep sea? Well, it's very likely in shallow waters that are coastal that much of the selection of green is just the presence of chlorophyll and algae, right? You don't want to make blue because that's exactly the light that's being absorbed. Now, if you're in a place that has a lot of sediment or a lot of mud being stirred up by the currents, one idea was that light just scattered from particles is wavelength dependent, so you're always going to favor longer wavelengths. But because of these existing mechanisms of shallower species of retaining a GFP and associating that with the bioluminescence, they're probably just moving into the deep over millions of years. So to wrap all of this up, in corals, the function of light may depend on the predator, and it doesn't have to be mutually exclusive. Because if you have opportunistic or you have specialist predators, it may serve as a warning signal for one or a burglar alarm for others. And then we were considering that turbid environments may be a strong selecting factor for green light. And again, if anybody knows about a visual spectra database, please give me a call. So I want to thank my co-authors and the funders. I think all my co-authors are here actually. So thanks, guys.
Brilliant. Thank you so much. That's fantastic. If there's time, if anybody has a short question, there's actually time to ask Warren. Yeah, Nico, green and blue are less absorbed by water and therefore can be seen from long distance, whereas be absorbed much faster. Could that indicate that there is an effect of the distance over which the bioluminescent signal is meant to work?
I would guess so. And that may be very short, but if you think of visibility in water, sometimes when scuba diving, it can be a meter or something. So I would expect that they might be right on top of each other when they're looking for it at night.
Brilliant, thanks. So we'll move on to our next speaker, Nikolai Hensley.
Yep. Move on. Okay. Good.
Great, thanks.
Okay, perfect. Hi, everyone. Thanks for coming to my talk. And also thanks to the organizers for all this. It's been really interesting thus far. I'm Nico. I'm a postdoc at Cornell right now, but I'm actually going to give you a rundown about my thesis work on the evolution and function of bioluminescent behaviors in Caribbean ostracods. Ooh, how does this function? Okay, there we go. So generally, I'm actually an evolutionary biologist, and I'm really interested in the predictability of evolution, whether diversification is repeatable, how many solutions there are to the problems of adaptation, and what kind of limits that ability for adaptations to evolve. But the traits I'm interested in are behaviors, and when we look at a complex trait like a behavior, this can be really challenging to analyze because there's lots of levels of biological hierarchy that contribute to these phenotypes. So we can kind of start at one level and assess, how does that contribute to the predictability of the behavior or the evolution of that behavior. But some people would argue that you don't actually need to know mechanism in order to understand that question. And I would say that in order to actually assess all the different kinds of hypotheses to explain about why a behavior or any phenotype exists in its current state, mechanism can be really informative to test alternatives like the predictability constraints that behind the evolution of the phenotype. And so to kind of get at this question about how predictable or how constrained a phenotype is, we can look at the genomic mechanisms of a behavior and try and see what correlates there are at that level. So the behavior I'm looking at is in these Caribbean ostracods. So these are small millimeter-sized crustaceans that use bioluminescence for anti-predator defense, and we actually just dropped that preprint online by Ellis and Goodhart et al. If you want to see it. where they've certain species in the Caribbean have actually co-opted this defensive anti-predator display into a mate signaling context. So these are just Caribbean species here, and you can actually see a time-lapse photo of what these displays look like. Males swim into the water and secrete discrete boluses of mucus that glow into these Morse code-like patterns that then females track and orient visually to. And so just for the rest of the talk, I'll be talking about mostly the diversification of these mating signals and our phenotypic correlates at the genomic level. And those will be annotated with this green, blue, and red. These are the specific genera in the clade. And then the outgroups, these non-signaling groups, will be in a lighter blue. And you'll be able to keep track of that with this little cladogram up in the corner. So as part of this process to understand phenotypic diversity and genomic diversity within these species, we kind of tried to assay a few different phenotypes of light production. The first one being color, which we've heard a lot about today. And so we looked at the peak emission spectra of little crushed up ostracods to see what the light looks like. And we see that it varied from about 450 to 460 to 470 nanometers, which isn't a lot. So we don't see a lot of variation, although there is some, and it does seem to have a phylogenetic pattern. So those from the clade in blue are actually slightly shifted to more green-blue wavelengths than the other clades. We also analyzed the decay rate of light, that is how fast light is produced in our 2019 paper, and we see that there is a phylogenetic pattern to this as well, or at least about the variation amongst the species in how fast light is produced. And then if we look at a more gross kind of phenotypic level at pulse duration between species, we see that different species can have very different pulses and how long they're visible for, anywhere from a few or 100 milliseconds from the shortest ones here to maybe 10 seconds or so. So we can see that there's a lot of variability between species and what these signals might look like. And the question is, how did they change? And what are the mechanisms governing that change? And so we've actually shown that at least two of these things are correlated. Courtship pulse duration is really related to how fast the light decays, which would seem kind of obvious, but this is a really important starting place for us to try and connect the molecular mechanisms to these phenotypic measures that might actually be under selection and investigating that process. So this is a really great system to ask about the predictability of diversification and the mechanisms of diversification, because we have candidate gene, luciferase, that these animals have independently evolved, and they secrete into the water with a high energy substrate, which becomes oxidized and releases blue or blue-green light to attract mates. And we can express this protein in vitro, which is also a part of the work that we've done. So we've taken it and we've expressed it in 3 different cell cultures: silkworm cells, human embryonic kidney cells, and yeast cells. And here I'm just showing you the same ortholog expressed in 3 different expression systems. Either gray or white is our negative controls, and in blue is with the protein and the addition of the substrate. You can see a statistically significant increase in the amount of light upon the addition of the substrate. The axes don't translate across all 3 of these. I'm just kind of showing you the same data from the same ortholog, but we've been able to express different orthologs from different species in all of these expression systems to show that there's a really functional assay that we can then test mechanisms of, of light production. This is like the most I've talked in a while. Okay, so to get at this question of signatures of selection and relationships to phenotypes, we took transcriptomes from our species. We pulled luciferases from them, using phylogenetic bioinformatic methods. And then we used hyphae to look at patterns of selection across the codons. And then we also have this really powerful mutagenesis screen that previous researchers have done to try and connect phenotype to genotype, in this case, in a statistical method. So we haven't been able to do mutagenesis yet on these proteins, but hopefully with this analysis, we have places to now start doing that kind of work. And I'm going to run through just some of our more interesting results. There's a lot here, and we don't have time for all of that. But we can see that there's many sites expectedly under negative selection, probably to preserve protein function. And there's quite a few here in green that we see that are under episodic or pervasive positive selection. That is, they've diversified across the clade concomitantly with kind of the diversity of the signals that we see amongst the animals. Of note, we have, if we're going to look at, say, a phenotype and try and understand the evolutionary processes that are contributing to the diversification of that phenotype, we'll just take color to start. And we can see that many different sites under many different evolutionary processes are actually contributing to variation in color significantly. So there are sites that are neutrally evolving, there are sites that are under negative selection, and there are sites that are under positive selection, and all of those have functional effects on the color that we see. So we can't just say that maybe shifts in color are adaptive per se, because there are many sites that contribute to variation in color amongst these species that are also under different evolutionary processes. So it really takes, but with this kind of assay, we're hoping to be able to get down to some of those more functional questions, or we can test those hypotheses by expressing these proteins and mutating them to see how that might affect their function, change their color, something like that. One of these sites that's really interesting is this site 404 here. This is actually in other screens an N-glycosylation site, and we can see it's under strong negative selection, where mutations at this site in vitro show a reduced light output, but this acts epistatically with neutrally evolving site 375. And so this epistatic interaction may have consequences for the evolution of this function of this protein and therefore the enzyme itself. And we actually do see a replacement from an N to a D at this residue across the clade, suggesting that there could be some evolutionary significance to a change in the epistatic interactions in this protein. We also have this site here, 160, which is kind of my favorite site, which might be a weird thing to say. But this site, it was actually statistically correlated with both changes in the decay rate of light as well as the color. So we see that it's significant to two different phenotypes of, or functions of the enzyme. And so this might indicate that this residue has some kind of epistate or pleiotropy, where it affects multiple functions, and therefore it can actually link different kinds of selective pressures over evolutionary time. Okay, so that's just the selection at the molecular level, but it doesn't give us a really satisfying idea of how this protein actually functions and might influence courtship or any other kind of phenotype we're measuring. And so what we did was we did Caylus Menten kinetics on 5 orthologs from 5 different species by taking the purified proteins and measuring their output ability at different concentrations. And from there, we measured the specificity constant, which is a measure of the enzyme efficiency to produce product from substrate, and product in this case is light because it's bioluminescence, as you all know. And what we find is that there's a positive relationship between courtship pulse duration measured from the phenotypes in nature to the specificity constant. That is, the more efficient your enzyme is at producing light, the longer your courtship pulse durations can be. And so this gives us a really satisfying kind of a molecular assay to then start to ask questions about how pulse duration is actually made in nature, and we can measure that in the lab. Okay, so I'll skip that. Lastly, we want to know the function of this signal and how that relates to what we see out in nature, the variation out in nature. And so what we did was these playback experiments to virgin females of 4 different species, where we showed them 2 different LEDs that differed in 1 or 2 different patterns. And then we just asked, how do they behave using deep lab cut to track their motion? And I'll just show you very briefly some of these results. So we did 2 different things. We either changed the timing between pulses in these LEDs or the duration of the pulses to see how it affected female swimming behavior. And when we changed the timing between pulses, the IPI, we saw that females didn't really care about that. They didn't swim any faster, which might be a metric of their motivation to do mate searching. But when we changed the pulse duration, we see that females significantly increased the rate at which they swim, suggesting maybe that they're looking for mates, and that variation in pulse duration might actually be really significant for female mate searching behavior. So altogether, this provides us with the ability to go from a candidate gene to selection pressures in the wild, and kind of full circle it by changing things at the amino acid level, testing them at the behavioral level, and try and get an understanding of how selection might be changing phenotypes at the genomic level. So if you want to read about some of that, there's a paper on molecular ecology, thanks to co-authors. And that's it.
Thanks very much, Nico. That was fantastic and exactly to time. So we'll move straight on to our next speaker, Bridget Vincent.
Hi, everyone. I'm Bridget Vincent. I'm a third year in the Oakley Lab at UCSB. Thank you for coming to my talk. And today I'll be talking about the project that I've been working on over the last year, which has been estimating the origins of photophores in cephalopods. And so when we start to study complex traits, we can think about them in different ways. Also, this figure was made by my co-author and lab mate, Emily Lau, who is the next talk if you want to learn more about her work. So back to complex traits, we can think about them in different functional modules. We can think about, say, in bioluminescence, the production of molecules, the maintenance of an organ, behavioral control, catabolism, storage of these molecules, and then modification of that emitted light. We can also think about complex traits in the different levels of, or different biological levels of organization. So looking at these, for examples, we can study convergence with a perspective focused on organs. So just looking at glands, photophores, granular light organs, and then we can move to a lower level, such as cellular or subcellular, looking at those specific photocytes, or reflectors, chromatophores, all of those kinds of accessory cells. And then we can also look at photophores or convergence in these different molecules that actually produce the light. I'll be focusing on cephalopod photophores, which are found in a bunch of different species, including squid, cuttlefish, octopus, and the vampire squid. This first one's the firefly squid, and if you just wanted to see all the photophores, they're in my Zoom background. And they use their photophores for communication, for catching prey, and just a bunch of different environmental uses. And so when we look at cephalopod photophores, there's two main types. One is the bacteriogenic photophore, where we have a little crypt housing the bacterial symbionts that actually make the light. And there is usually only one main morphology that we see in the animals that have this kind of organ, versus the autogenic types of photophores, where instead of having that bacterial symbiont, we have photocytes, where the animal creates its own light. And here we can see many different types of morphologies in the species that create this type of light. Previous students in this lab have studied the convergence of photophores at the organ level. Sabrina Pankey was looking at sepiolid squids and found that there's at least 2 independent origins and possibly a third, but we wanted to dig a little bit deeper and ask if these patterns of convergence remain consistent across biological levels when studying these photophores. And so to tackle this question, first we needed to construct a new phylogeny using recent data, then determine how many times bacteriogenic photophores, autogenic photophores, and the accessory tissues of autogenic photophores originated, and then assess whether all of these components follow a similar pattern. And so when we're looking at previous phylogenies of cephalopods, Sabrina Pankey, the study that I mentioned before, had 70 species with both transcriptome and nucleotide data. And then Lindgren had 18 species and was mostly looking at how transcriptome sampling can help with phylogenies. Sanchez had 124 samples looking at the genus level with only nucleotide data. And we are including 371 species with both transcriptome and nucleotide data. And so we took single gene data from NCBI using 13 different genes for these different species. Not every species has all of these genes, but we are using 13 total loci. And we also included transcriptome orthologs from Lindgren and Tanner, using math to create alignments, then using IQ-tree to create gene trees. And then for the tree that I'm showing you, we used a straw to create a coalescent tree. And so here is kind of a summary of our phylogeny. Our cirrate octopuses are monophyletic, sepiids are polyphyletic, and then some egopsid groups that have kind of been argued about in our past phylogenies have been found to be monophyletic. There are a few odd placements that just happened with our data. So Vampiroteuthis is in kind of a weird spot, not quite sister to all of the decapods. And then there's a few odd little weird species here and there to look further into. But these species have not had any or have only had little effect on our number of origins, so it didn't really affect our conclusions. Going back to our different photophore types, we did notice that there are a lot of similar core tissues. So both types have this photogenic region, they have a lens, pigment layer. For the bacteriogenic ones, the ink sac functions as the pigment layer. And then there's also this reflector. So when we looked at autogenic, we wanted to dig deeper into that tissue biological level to see if convergence patterns remained. So when we were characterizing our different species photophores, for bacteriogenic photophores, we scored presence or absence. And then for autogenic, we scored presence and absence and then separated those organs into type 1, 2, or 3 based on some previous definitions. And then we also characterized the presence of the lens, color filter, reflector, and iridosome, as well as the different types of those. The lens, color filter, and reflector give us some insight into the tissue level of organization, and the iridosome gives us a subcellular perspective. So looking at our bacteriogenic origins, we found 7 in this sepid myopsid clade. And then for autogenic, we found 17, 18, and 11 origins respectively for those 2 types, most of which are in the agopsid clade, with a few in octopus, and then a little bit in the sepids. For our lenses, we found 3 origins of type 1 and 1 of type 2. Color filter, we found 5. Reflector, we found 1, 13, and 5 origins. Again, we have this agopsid groups and then some in the sepids and our Vampiroteuthis. And then for the iridosome, we have 4 origins of type 1 and 16 of type 2, both of which are found in these 2 main groups of origins here. And so in the future, we are planning to compare coalescent and concatenation approach for creating a phylogeny, adding gene and site concordance analyses, and then also adding an independent origins test, since we know that we likely have an overestimate of these origins, also probably because we have some missing species data. So some of these origins are just single species popping up where we don't have a lot of sister species data. And so we have created a cephalopod phylogeny, found that light organs and tissues are convergent across agopsid and sepid lineages. And because we looked at those iridophores, we can see that there are probably different strategies for forming some accessory tissues at the subcellular level, because those 2 different kinds of iridophores are defined by the different materials that are used to create them. And I would like to thank the GRFP for funding, all of the Oakley Lab members, past and present, our department, and the people at the Scientific Computing Center who have been a big help in learning how to do all of these different methods. Also, if for some reason you need some squid images, we have some new ones at Memento Animalis. So thank you all for listening.
All right, thank you very much, Bridget, for that really interesting talk. We have a couple minutes, actually. So are there any questions for Bridget? I'm really interested in the structures of the photophores. What, perhaps you already talked about this, but at what point do those 2 structures diverge in the gene trees?
At what point does it separate into autogenic and bacteriogenic?
Yes.
Okay, so there isn't really a point where both of them come from a common ancestor, so they each originated on their own. But we do see both types in the sepid lineage, and we really only see the autogenic ones in that agopsid, like the big, huge squid lineage.
Okay, thank you.
But the autogenic ones are also found in octopus. So there's a lot of independent origins for these different morphologies.
All right, thanks. Are there any more? Anybody have any more questions? Okay, well, with that, if Emily's ready, we can move to the next speaker. She'll be talking about some of the same things. So thank you very much, Bridget.
All right, hi. Can everyone see the presentation?
Yes.
Okay, cool, thanks. Hi, everyone. My name is Emily Lau, and I am a PhD candidate at the University of California, Santa Barbara. And today I will be presenting results from one of my thesis projects, which is on the bioluminescent toadfish Porichthes. I am particularly interested in convergent evolution, which is when similar traits independently evolve. And long-standing question is, do convergent traits evolve from conserved or disparate trajectories? Now we know that they can evolve from both types of trajectories. So we can tackle questions like how and why do convergent traits evolve from different or similar trajectories? Bioluminescence, the biological production of light, is really great for addressing these questions because it convergently evolved at least 90 times with organisms that produce light by themselves, which is called autogenic bioluminescence, or with the help of symbiotic luminous bacteria. Because it is a complex trait, phenotypic convergence might not extend across biological levels. For example, these 3 organisms, an ostracod and 2 fishes, produce bioluminescence using radically different organs, but use the same small molecule, bargulin, in their bioluminescence reaction. An excellent example of convergence at an organ level is the independent evolution of lensed photophores, light organs. Though they evolved independently, many are comprised of similar structures such as reflectors, pigment, and the lens, which is all circled in green here. So the question that we are addressing here is, are photophore lenses convergent at a molecular level? And generally, we know very little about the molecular basis of photophore lens, but we know that the photophore lens of luminous squid contain these structural proteins called crystallins that are found in the lens of eyes. The photophore lens of toadfishes, however, they have a novel crystalline that is not found in their eye lens, and it's completely non-homologous to any known crystalline. So here we aim to answer, what is the genetic identity of this novel crystalline, and how did it evolve? To investigate the genomic basis of crystalline evolution, we assembled 2 hybrid de novo genome assemblies for Porichthes myriaster, which is a luminous toadfish, and Aphos porosus, which is the most closely related non-luminous toadfish. And we combined our genomic data with exon target capture data, and we generated a molecular phylogeny of toadfishes. All toadfishes here in gray are non-luminous, and only the Porichthes species are luminous. And we found that Aphos porosus, which is non-luminous, was actually nested within the clade of Porichthes, which suggests that this was a loss of bioluminescence. But luckily, for our analyses, there is a publicly available genome for the closest outgroup, the Lasoprene. And using partial sequencing data, we identified the novel crystalline gene and searched for similar genes in public databases. And we found similar genes only in other bacteria, fungi, and other fishes. And these genes are essentially functionally uncharacterized and unclassified. So we proposed that the gene originated from an ancient horizontal gene transfer event. The alternative hypothesis is at least 10 independent losses shown in this tree. So the most parsimonious explanation is 1 horizontal gene transfer event instead of 10 losses. And this horizontal gene transfer event provided the source of genetic material that evolution could tinker with. So we found that the crystalline evolved through gene duplication events that are specific to luminous toadfishes. And this is a phylogeny of crystalline-like genes that are found in the genomes Porichthes, the luminous toadfish, Aphos, which lost bioluminescence, and Lasoprene, which is non-luminous. And only the luminous species and the non-luminous species that lost bioluminescence have genes that contain the amino acid sequences previously reported, which is in this gray box here. And so this supports that this novel crystalline evolved only in luminous toadfishes. Furthermore, we found that the crystalline genes in Perichthys and Aphos are found really close to each other in the genome, which suggests that these tandem gene duplication events was responsible for this expansion of crystalline genes that we're finding in Perichthys. And to summarize, unlike squid, the photophore lens of Perichthys had this novel crystalline that's not found in its eye. So what is this gene and how did it evolve? Our results suggest that a horizontal gene transfer event provided material for evolutionary tinkering, and this was later co-opted during the evolution of this novel crystalline. So in addition to gene duplication, which is really important, an ancient horizontal gene transfer event was crucial for the evolution of this crystalline. And we are currently exploring the evolution of other genes that are found in the Perichthys photophore, so stay tuned for future results. And thanks for joining my talk, and a special thanks to the organizers, the Oakley Lab and collaborators. Please feel free to reach out if you have any questions about this work or if you want to talk about toadfishes. Thanks.
All right, thank you very much, Emily. We are a bit early, so if there's any questions, you can type them in the chat or raise your hand. And let's see. Okay, a question. Here we go. What is the source of the horizontal gene transfer?
Okay, cool. Thanks for that question. I'm pulling up the horizontal gene transfer tree again. And so it's either bacteria or fungi, and we can't pinpoint which one unless we do further genomic phylogenetic analyses to really figure out which one is sister to the rest of the fish ones. And is this sort of horizontal gene transfer unusual, and is it associated with other traits? I would say that this sort of horizontal gene transfer, leading to novelty, is sort of becoming increasingly common that we're finding. For example, there is the whitefly that through a horizontal gene transfer event obtained a gene that from its host plant, which allowed it to become resistant to a toxin, I believe. And so it's becoming a lot more common now. I know that horizontal gene transfer was originally in the past thought to be super rare, but with these genomic advances, we're seeing that it is a lot more common and there's more strong support for it.
Here's one. How much is the novel crystalline in the lens? For example, what % in weight?
The novel crystalline in the lens is the major predominant soluble protein. So it's through Dove et al. In 1993, they did sort of like an SDS page and protein extraction for the photophore lens, and they found really only a handful of proteins that are found in there. And this crystalline was the most soluble, was the most abundant in the soluble fraction. Okay, aren't toadfish benthic? So why would they have photophores on their belly? So actually toadfish come up to the shore around summertime where they actually dig out these nests and they mate. And so they do come up close to shore. And also it's hypothesized that they do sort of migrate and feed on animals throughout the night. So potentially they have photophores on their belly that work for counter illumination to sort of mask the light that is downwelling. Thanks. Yeah, it's super exciting actually. They come up to shore around Santa Barbara too, and San Francisco, I believe, like way back in the 1900s, they were super loud. So they make these really crazy noises. And so everyone around the ship area, the dock, thought that, there was something weird going on, but it turns out there was just all these toadfishes that are singing. Yeah, are there any physical characteristics that you can use to compare the lens crystalline and the photophore crystalline? And do they do the same thing? Yeah, we can definitely do sort of these protein extractions from the lens crystalline, the lens of the eye and the photophore. And you can sort of compare the size of the proteins and do protein sequencing to figure out if they have the same amino acid sequences, if that answers your question.
So you're saying both the lens crystalline and the photophore crystalline serve the same functional purpose as in their being in the lens. So they're creating a lens that is transparent and that helps with some kind of light direction.
Yeah, I would love to know, the structure of the protein, but it's completely uncharacterized, so you can't even really model it based on homology. Yeah, I think the next step would be actually figuring out the physical properties of the protein and seeing how it can achieve the same as the crystallines that are found in the lens of eyes.
Okay, if we're done, we could move early, a little bit early to Avalon if Avalon's ready.
Yes.
Okay, yeah, let's go ahead and move on. Thanks a lot, Emily.
Thank you.
Yeah, thank you. And thank you everyone for going a bit quickly. I was a little nervous about time, so this feels great to have a few more minutes. I won't push it too much. Can everybody see my screen okay? Yeah. All right. So yes, thanks everyone. So I am a very late stage PhD student at Tufts University, and I've studied fireflies and light pollution for the past 7 years. But today, my main purpose is to convince you all that fireflies are an incredible study system for thinking about living light. They are literally living light, so it should be pretty easy, I hope. So let's start with by talking about what fireflies are. Fireflies, of course, are insects that come out in the month of June in the popular Nintendo game Animal Crossing. And when you take them to the museum curator, Blathers, he's a little upset because they're insects, but then he'll tell you that fireflies are beetles that produce bioluminescence via a chemical reaction housed in their abdominal lanterns, and that males flash about in the night sky to attract females while their larvae turn on the glow to put off predators. And Blathers does a great job. There's not all that much that I can add. Fireflies common in North America and Japan are typically communicate in this way. The males will float around in the air emitting advertisement flashes, and the females waiting on the ground below will flash back when they see a guy that they like. But there is a little bit more detail I want to discuss before we dive in, so I brought Blathers back to give us a few more tips about fireflies. The first one is that fireflies are super diverse and species rich. There are over 2,000 species on every continent except Antarctica. Here in North America, you often find up to 15 species of firefly in the same spot. And each species will have a unique pattern of flashes, a flash pattern that they use to distinguish themselves from others nearby. The other thing is that fireflies are fearsome predators. They are universally carnivorous. The larvae eat earthworms, snails, and things like that. And some adult fireflies actually eat other fireflies. So here is a big firefly in the genus Photurus snacking on a male of the genus Photinus that has been lured in by her flashes. And if you want to learn more about this, I really recommend this video by Deep Look. And then the last thing that I think everyone should know about fireflies is, of course, that fireflies match their bioluminescence spectrum to the peak sensitivity of their long wavelength photoreceptor to optimize detection of conspecific signals at twilight, naturally. So basically, the ancestral insect configuration for vision, I'm sure many of us are familiar with it. Insects tend to have a UV, a blue, and a green photoreceptor. Fireflies, like many other beetles, have lost their blue photoreceptor, so they're left with a UV and a green. And UV they use for whatever they need to, but the green has a very specialized function. It's basically optimized to detect conspecific bioluminescence, which looks, I kid you not, like this. It matches almost perfectly, 1-to-1. So they use this photoreceptor to see others of their species. And the story goes something like this. So the original fireflies were fully nocturnal, and they see green and they flash green, and green shows up really well against darkness as modeled here by a spectrum of starlight on a moonless night. But then some fireflies, like this Photurus over here, started eating other fireflies, and all of a sudden the night was a very dangerous place to be. And so the prey species, Photinus, shifted earlier in the evening. So they're now, at least some of the species are crepuscular or dusk active. And they were then faced with a bit of a visual ecology conundrum, which is green flashes don't show up super well against green trees and leaves and what have you. And so they shifted the color of their flashes to yellow to maximize their contrast against the green of leaves and the bluish light of twilight. So this is the basics of Seliger et al.'s model for the optimization of signal to noise ratio for the detection of bioluminescence by fireflies during twilight, which is actually a really great read. I recommend it to all of you. There are a couple more details about it though, which are, before I'll say it with my words. So basically, you can't just turn from green to yellow though, because if a firefly shifts its bioluminescence spectrum away from its green photoreceptor, it's going to lose visibility. So instead, the first thing that fireflies do is they shift their vision using filter pigments. So here is a crepuscular firefly. Its green photoreceptor might originally have looked like this curve here, but through the addition of some pinkish filter pigments that cut out a lot of the green wavelengths, they're able to sort of narrow their vision to focus in the yellow region of the spectrum. And at first, they're losing a little bit of the visibility of bioluminescence, but they're cutting out a lot more environmental light, so it's still a pretty good thing to do. Yes, okay. So this is such an elegant hypothesis, and it is backed up by some data. So these are electroretinograms collected by Abner Lal and colleagues a while ago. And on the left, these three graphs are all crepuscular firefly species. You can see they have a peak sensitivity of around 575 nanometers, let's say, which is pretty yellow. And then over here on the right are two nocturnal fireflies with peak sensitivities right at 550. And then down here, this is a day active firefly I just put in for fun. So wonderful. We have 5 data points and they look great. There's just one small issue, which some of you may have noticed. All of these species on the left are the prey species of firefly, they're all in the genus that are preyed on by these fireflies here in the genus of predatory fireflies. So these are very, very different animals, and they're flashing for different reasons. They have very different behavior. And so it's kind of an odd comparison. And we could make a much better comparison by picking a species of Photinus that comes out later in the evening. But nobody had done this until the summer. So I was able to collaborate with Damon Crook at the USDA facility in Buzzards Bay. I shipped him a whole bunch of fireflies because I couldn't go in because of COVID. So they went through the mail, a whole bunch of Photinus obscurellus fireflies, which are in the genus Photinus, but come out later at night than all of the species that we've seen so far. And we were the first to get a non-crepuscular Photinus species, and also the first to get the spectral sensitivity of a female Photinus firefly, which haven't really been studied all that much. And you can see the relative sensitivity of the UV and green photoreceptors are quite different between the sexes. This could be an artifact of the time of day that the data were collected. I'm a little more interested in where the peak of the green photoreceptor is located. And so in order to compare, I put the males and females together, and then am comparing them to our crepuscular Photinus and our nocturnal Photurus. And you can see that actually it does seem to follow the pattern pretty well. So while Photinus pyralis is about 575, Photinus obscurellus, I would say, is around 555. or at least that's what my generalized additive model told me. I will be investigating this more in the future. So even though I was a little skeptical of this hypothesis, it does bear out when we have more data, which is really exciting. Okay, now here's my actual talk. So I want to talk about light pollution because it's this very interesting visual disturbance to the nocturnal environment that's totally unprecedented in terms of evolutionary history, and also way more widespread than you might think. One recent study showed that in the past 25 years, the night has gotten 50% brighter. So this is something that animals are dealing with all the time. And there's many ways to think about light pollution. I'm going to talk about sky glow, which is this sort of reddish haze that you see over cities that sort of uniformly changes the background. So if we go back to our little model and add on some light pollution, you can see it is indeed very reddish. It's brighter than twilight in a lot of cases. It makes the twilight last longer and all these things. And so looking at the optimization model, I began to make a few predictions about how fireflies might deal with this stressor. The first is they might increase the conspicuousness of their flashes, and they could do this in a couple ways. They could make them greener to contrast more against a reddish sky glow background. They could make them brighter. Brighter is always better as far as visibility goes. Or each individual flash could be longer, or maybe they could put more flashes into each flash pattern or more flashes per minute. The alternative is failure to communicate, courtship failure, failure to reproduce, and all of a sudden we have a conservation crisis on our hands. So can fireflies become greener in light polluted areas? The optimization model tells us that they shouldn't really be able to do it on an individual level. This is something that happens over evolutionary time through changes in spectral sensitivity. There's very little data on this, except this amazing study from 2016, which shows that the firefly species Phytinus pyralis actually does flash in a range of wavelengths that are sort of unique to each population. So each population has its own slightly different color. I've exaggerated them on this map, but it's a difference of about 10 nanometers. So, you know, why are they doing this? Well, we know that it doesn't have to do with whether the habitat is open, mixed, or closed. We know that it doesn't have to do with latitude or elevation. Could it have to do with light pollution? I'm still working on that, so I'm just gonna keep moving, but hopefully we'll be able to revisit this soon. Okay, how about brighter, longer flashes? If a firefly could flash more brightly when it was light polluted, that would be great. Just the way that birds sing more loudly in urban areas. It would be a very classic example of adaptation. So to study this, I worked with a Taiwanese firefly species during my master's and set up this unique contraption where I was able to put a spectrometer probe right up against the abdomen of one of these fireflies and record their flashes in real time as I changed the lighting overhead. And so it produced a graph that looks something like an EKG, where time is on the x-axis and each peak is a flash. And you can see in darkness, the flashes are all at about the same intensity. When you add a dim green light, all of a sudden they get much, much brighter. And each individual flash also lasts a bit longer, which is what we would expect. When you take that light away, flashes gradually fall back to their baseline level of brightness. And when you add a really bright green light overhead, again, the flashes get brighter, but now we start missing some. There's sort of these competing responses of flashing brighter and then maybe just giving up and not flashing at all. So I'd be happy to talk more about this later, but let's keep going. Can fireflies flash more flashes, or are they doomed, let's say? And so to study this, I've looked at fireflies engaging in dialogue in the laboratory under different levels of light, found that males flash less overall when lights are overhead, and females flash way less than we knew. And it's pretty dang concerning for the future of fireflies. But still, there's a lot to be learned. So let me just wrap up by saying that it does appear that fireflies can attempt to adapt to light pollution somewhat, but if we really want to be careful and protect these insects that we all love so much, we should do our best to limit the amount of artificial light out at night. And in this spirit, I partnered with the Xerces Society to create a set of lighting guidelines for fireflies, which I'll put in the chat afterwards. And I just want to thank everyone who has funded me and everybody here for listening. And look at this horrible photo of this nature reserve by my house. We got to stop doing this because fireflies live here, but I don't know how much longer they will. Thank you.
All right, thanks a lot, Avalon. Perfect timing. We are pretty quickly going to move on to, so everyone submit your questions or give your questions to Avalon. I think the chat will end in this break room when we leave it because we're going to go see the keynote speaker now. So you might contact Avalon separately or in the other room. And thanks a lot. This concludes session.